Hello, I'm Will. Welcome to ResearchPod. Synthetic biology, as showcased by Stephen Freeland's work at UMBC, is revolutionizing future research. It redefines biological systems for groundbreaking applications in biotechnology and medicine, promising to solve global challenges and unlock new frontiers in science. Or at least that's what ChatGPT has to say about it. We do indeed cover many of these aspects of his work in our discussion today, but also how generative tools like digital AI are changing the wet lab experience of doing research, plus the importance of public domain in owning, understanding, and advancing all the many ways the future is taking shape. Professor Stephen Freeland. Hello.
Hello.
Right, thanks so much for your time today. Could you tell me and everyone listening to this a bit about yourself, your background?
Sure. I mean, the older one gets, the more one has too much to pick from. So guide me here. But I'm a biologist. I think that's probably the most important thing to say. I'm a biologist in my heart and in my soul. And I'm not saying I chose it. I'm saying that somewhere during school, it just seemed to be both something I was better at than average, but something I just enjoyed. And I won't kid you, there are years that go by if you choose the academic route where you hate what you're doing. But I've never stopped loving biology, right? So that might be the most important thing to tell you. What else would you like to know?
Out of anything, why biology and not plumbing or...
Why biology? Isn't it fun? You do something like this and people ask you the questions you never get asked that make you think. Let me try. As you're talking even, my reflection is probably that firstly, it was pragmatic. I was doing well in it at school and I wasn't doing well at other things. I suck at languages. Thank goodness I was born English, given that that's the world, the scientific world's language. Because I probably couldn't do this otherwise. I know it'd be a lot harder. Anyway, I was good at it. So there's positive reinforcement. That's the practical answer. But to give you a soulful answer, yeah, I guess that if I think about it, I was raised in the countryside and raised to believe very much or to see the beauty of nature, right? Sounds hokey, but it's never let me down. And by the way, nature is ugly and cruel and hard, but there is a savage beauty there, and there's just something that draws me. And then perhaps at this stage, third of the answer, it's true that there's so much that's unknown. I mean, a lot of that has changed in my lifetime, but I would say go into a science where there's so much that is unknown because you stand a real chance of finding out new stuff, right? And that's a big draw with biology. I want to give them a shout out here. The University of York in the UK, very analogous to the university I now work at. But it was sandwiched for me between Oxford and Cambridge, and both were wonderful, both have extremely fine minds, but University of York changed everything by taking on, way ahead of its time, a small crew of biologists each year into a master's program to teach them fundamental mathematics and computer science. Now, I'm a bioinformatician, have been ever since. Fancy way of saying I use computers to ask biological questions. They were so ahead of their time, and it still inspires me that a smaller, humbler university can wisely find something that is forward-facing and beat Cambridge and Oxford and everyone, frankly. So there you go. That was a big milestone. Learning that you can use computers and mathematics to study at least part of the beauty in biology. Enrollment in biology degrees is declining. Computer science exploding. And we can speculate all you want, but the answers are probably pretty obvious. I think that matters. I think I want more biologists, right? There's so much there and so much we don't know and so much potential. And often we think about that biomedically, and heaven's sake, I'm glad that we have biomedical, but there's so much more. Origins, understandings, products, manufacturing. I want more biologists.
And on a personal note, are you prone to kind of poetry, philosophy, looking out the window and getting all wistful about it, or are you more of kind of a boots on the ground, get in the lab and get things done kind of guy?
Yeah, I'm probably more like the philosophical poet than I'd like to think. But yeah. I guess that is a driver, isn't it, all the way through? I mean, that's what it does for you. If in childhood you love nature and you choose to develop that academically, that's going to be with you a lifetime, isn't it?
What we talked about in some of our setup calls to this is framing things as lies to children. And that's possibly unkind because it's never really a lie to tell people what it is they are capable of processing and dealing with the rest of the context that it sits in. So to start off from a point of DNA, can you tell me and people listening, what is DNA and how is that relevant to, I don't know, let's say a 10-year-old, a 15-year-old, a 20-year-old, and what are the layers of understanding that we add on with every stage, key stage of learning, or like whatever the American grading system comparator is?
Right. Good. That actually helps me. That last little twist helps me with a very big question. where would you begin? Let's begin with something that maybe everybody already knows, just to agree where we're starting from. DNA is genes, it's genetic material. And genes program life. They are the instructions that you decode to make life. And it's interesting, already I've got a you there. That's the beauty of it. There's no you there. That's if they decode themselves to make life, right? Okay. Yeah. So that's the 10-year-old thing, and it's important to know because you will at every grade. I think these days you would be taught something of that to understand the power, good and bad, that exercises over your life. You are genetically predisposed to certain medical conditions. You also have genetic predisposition to certain things that you're good at. Much harder to measure, by the way. Um, it's not everything, and that's where a lot of the debate lies, nature nurture. But that's what we need to remember: is genes program life. We don't know how or what it even means to be alive without genetic programming. That's part of our fundamental understanding of life, right? So you ready to go up a level from there, or do you want to ask anything about that? Okay. I think the next thing I would want to teach you is that DNA is a polymer, which is just a fancy way of saying it's a lot of building blocks linked together. And right here, let's introduce an analogy that I think will take us surprisingly far in our conversation. It's like words are built of letters. DNA is built of things called nucleotides. They're the building blocks. There are only 4 of them. They are the famous ACGT, and it's all a question of what you spell out with them. And you think, well, there's only 4, but then you think, well, there's only 26 English letters, and that's suffice to bring us all of modern literature, fiction and non-fiction, right? And it's like, oh, there's power in those 4. Yeah. So that's the next thing. It's a polymer built of those 4. And the polymer means that you link them together so it becomes one big molecule, chemically speaking. But it's the sequence that we've just said, like it's the sequence of letters that make a word. So DNA is a sequence. And we've heard that, DNA sequencing, right? So that's probably layer 2. I want to think hard, but my temptation is to leapfrog you now very close to what I do with layer 3. I think you do a biology degree, you're going to find out 15 layers I've just skipped over. But the next thing I want to tell you is that most biologists, and I would hazard a guess ChatGPT, would tell you wrongly that there are only 4 letters.
So we've set the bounds, 4 letters. That sounds pretty comprehensive. That sounds like a good structure to work within. So why is it not true?
Because it all depends how you meant the question. And if you want a philosophical theme to resonate throughout the rest of our conversation, that would be it. It all depends on what you meant by the question. Boy, if you ever go into computer science, remember that for me is everything in computer science. We call it GIGO, garbage in, garbage out. Less formally, Hitchhiker's Guide to the Galaxy. They built a supercomputer called Planet Earth to figure out the meaning of life, the universe, and everything. And after 4 billion years, it concluded the answer was 42. They'd asked the wrong question. So let's go there. If you mean it as a geneticist, it's true-ish in the sense that life builds with those 4, all of life, right? That we know of. And to be fair, there was a very big splash, flash in the pan, I want to say 3 years ago, for finding that it was viruses, wasn't it, that have a 5th letter. But to be fair, they've sort of changed one of the 4. But I wrote a little opinion piece on the back end of that to say, you know what, that is the final nail in the coffin of this whole idea of not only 4, but of other building blocks we'll talk about. That's just the empirical proof, because biologically there are 4, but we know that there are hundreds, if not thousands, of derivatives, we tend to call them, like, they're not those chemicals that are used in cells. They're just not used to spell out genetic messages the way we preach it. And I'm thinking, everything I say, I'm like, no, that's wrong. Right? Some forms of derivative are used to control which genes are expressed, which ones are decoded. So, in a sense, we are using those in genes, but traditionally we say, no, there are 4 letters that are decoded to build proteins, which then make metabolism, okay? But there are hundreds in a cell. There are, where should we go next? Chemically, they are infinite. It's a class of chemical. You have to adhere to certain rules, but at least functionally, unless you introduce some sort of maximum size, you could have infinite nucleotides. So certainly the 4 we have sit in a theoretical space that is much larger, and biology demonstrates that space is useful and used. And then perhaps the final strand, because so much of this will echo amino acids when we get there, is that we've actually known for quite a while now that there are more than 4 that are formed abiotically by the universe for free outside of Earth. Traditionally, the way we've understood what is formed chemically is to look for a meteorite, which is just a word for space debris that lands on Earth. But you can take it into a lab and you can analyze it. And that plays a very big role for both amino acids and nucleotides. But for nucleotides, we've known that there are more than 4. There are tens. So I would suggest that all indications are that the four are an evolutionary outcome, not a requirement for life, and not even a truth about what life is currently.
When we say that meteorites contain amino acids, as we understand it, the
Mm.
building blocks of life, there'll be some people out there who will leap to the conclusion that, well, that means alien life, because if
Mm.
they have the
Mm.
right building blocks, then life is surely inevitable? Question mark.
There's several things going on what you just said. For what it's worth, let me just confess, I do believe that there should, in our current state of scientific understanding, be life everywhere. I'm not sure about multicellular intelligent life at all, but there should. So that bit. But yeah, the fundamental point here is we find the building blocks in meteorites. Well, isn't that a residue of alien life? No, I think there is good reason until further shocking evidence emerges, if it ever does, that to think these are formed abiotically. Um, I could give you some technical details, and maybe that's going a bit far fast, but I just say that there are various features of amino acids produced outside of biology that you don't get by amino acids. No, that sounds too opaque. There are mirror images of amino acids in particular that can be made. You're equally likely to make the left-handed form as the right-handed form, but life only uses the left-handed form. Whole other rabbit hole we could go down. Point is that in meteorites you find both in an equal mixture. That's one example of the sort of technicality. But these days, I could chase it through and say we even understand the reaction pathways, we understand how the chemistry is happening. So it's sort of like we've got the whole story on how these things are being produced by the universe without life, right? Maybe that's what I should have said. Right?
Well, let me put this question to you then. If we know that they can be formed abiotically, have we successfully made them on Earth? And if we are capable of synthesizing DNA on Earth, what does that tell us about the origins, possibly, of these molecules elsewhere?
Good. And I'm going to have to untangle a bit here because, like a bad sort of street performer, I accidentally allowed you to let us slip into talking about two different things. We were talking about nucleotides and DNA, and amino acids are what are the other half of it. So I want to go there first because the point is that with the exact question you've asked, you get very different answers for those two.
Let's try and work on the building blocks at their smallest resolution. So a nucleotide, that is an A, a C, a T, or a G, or otherwise, and it can be created using non-organic. It can be assembled using non-organic means. Assembled, created.
Because a nucleotide is a chemical structure that contains a sugar molecule called ribose linked to a nuclear base, which is actually the genetic letter, and to a phosphate. There's your chemistry. But those things are deeply challenging to make. We absolutely can, and for a long time, have known how to make nuclear bases, and those are the things we find on meteorites. It is highly controversial. A couple of groups in England believe that they have basically shown an ingenious mechanism to make a nucleotide, but without wanting to start a whole fight here, it's not consensus science. Um, if I paraphrase crudely, a lot of colleagues say it says more about their ingenuity and brilliance as chemists than it does about the abiotic universe. And we can go into that if you want. But that's so right there is nucleotides, no, nuclear bases, yes, and amino acids, yes. And we haven't talked about amino acids yet, but I do think it's significant that at that level you get those two for free almost everywhere. And it's nuclear bases where there are tens of them in meteorites, not just the four. It's very challenging to get them stuck onto a ribose and a phosphate, which makes a nucleotide, which is the building block of DNA. And it is exceedingly challenging to get those building blocks to link together, let alone to do so in a useful sequence. And somewhere in that, I've actually transversed from huge challenges of chemistry to huge challenges of mathematics. And we can talk about that if you want.
We're straying further and further from, capital B biology then.
Yeah. Yeah, but what does it tell you about life? I mean, just to sort of conclude the original question, and then we can dive into this a bit more. That's one of the big three lines of evidence that I think suggests to me that I'd be surprised if life wasn't common in the universe. The exact stuff at the core of what makes life on Earth is, we now would say, is given to us by free a lot of places in our own solar system, let alone the rest of the galaxy.
Well, if we have nuclear bases to make nucleotides, what can you do if you can make the building blocks of life?
So that is a different, intellectually overlapping field. It's a much bigger field. And the first thing to sort of hint that your question is an important one is it's a much bigger field because there's a lot more money flowing through it, primarily biomedical, but into sort of biochemical engineering. You know, the borders get boundary because there are so many useful things you can do if you start playing with nucleotides beyond the four. Now, there are whole classes that only begin to sort of like, well, a lot of them do this and a lot of them do that. But one thing is you can jam up life in very, very specific ways. So these things are very powerful for fighting viruses and cancer, right? And so synthetic biology, but honestly, they're good to jam up anything you want to study. You can almost sort of jam the machine and freeze it and then study it, which is often the enemy, is like it's happening too fast or whatever. That's one of their common uses for what would I say, non-canonical nucleotides would be one phrase that you could use for them. Another big class of using them is you can attach weird chemical groups to them that make it easy to find them later on. So you can sort of, like paint a target on them so that you can then figure out where they went to study processes that you're interested in, right? So those are just two examples of vast subfields that are very commercially and engineering oriented, right? That use these other bases. And I think one of the things we are rapidly learning to do in our smaller community that is interested in origins and astrobiology is to try to get much more serious about keeping pace with that. It's moving so fast, it's almost impossible to keep tabs on it. And it's not our field. But is that enough of an answer to get you going?
I mean, to think about things in kind of, to pass it in terms that I can hopefully keep up with, it's like an adhesive that you can stick things to it, but also it can fix things in place. If I think of
Yeah.
videos where I see people
Yeah.
doing carpentry and they flood the wood with resin, a synthetic, boat lacquer basically, to fix
Yeah.
the wood in place, and then they can shape it to their whim or put cool colours onto it.
I think, as a keen word worker myself, if that's what you take away from it, that's good enough. But let's go back to layers and layers. I would say that the first thing I would change about that metaphor, if you wanted a better understanding, is it's not like you saturate natural DNA with these things. It's more like you place them carefully inside the DNA sequence because you can find them, or because you know how that DNA sequence folds up and catalyzes a reaction, and you can place them at a specific position where the one reaction they do gets jammed midway, and now I can study how that reaction takes place. Does that make sense?
So you're going in there with a very specific wrench for the big gears
Yes.
rather than
That was
just
the
seeing
metaphor I was
mistakes.
searching around for. And think about that. It really is like a toolkit for studying biology at the molecular level.
What boundaries would you say have been set on the research that you are doing, and how do you see ways in which future research, 5, 10 years down the line, if there's anything that's a twinkling in some distant researcher's eye, might explode in new directions? Like, we can be as rosy glassed or cyberpunk glasses as appropriate about this.
Gotcha. So I'm going to be slightly cautious in what I say because for the first time in my scientific career, the truth about what is actually happening right now in Europe as a result of the work we're doing is scoopable and very poachable. And so I'll just be a little guarded, but we'll be fine with that. In fact, at this point, I think I must respectfully drag us back to this thing I keep harping on about amino acids and DNA very different. So, at a very simple level that we might teach high schoolers these days, you can think of the molecular basis of biology being two halves, two different chemicals wrapped up in a third. And the three concerned are DNA, proteins, and membranes. Membranes are made of lipids, and they probably won't feature again in this story, but it's those other two, DNA and proteins. When I talk about genes decoding themselves, they decode themselves into one very specific other chemical language. That is the chemical language of amino acids, which themselves link together to form a polymer, but that polymer is known as a protein. All of metabolism, which just means the network of chemical reactions that are a cell, is, to a first approximation, proteins. Proteins are the catalysts. There are also many of the structures that make up a cell. So when we talk about genetic programming, we mean programming to make proteins because proteins make the cell, but then the cell, at least from an evolutionary perspective, its primary purpose is to replicate the DNA, and round we go. That's life.
That's
Okay?
how viruses and bacteria work, is keeping that cell going as well.
Absolutely. And of course, that's what makes viruses so sort of interesting, and debates about whether they are life is they only contain the genetics. They need to hijack a cell in order to make proteins to complete their life cycle. So yeah, two halves, very, very important. conceptually, but also very important to understand chemically, unrelated languages. If anybody is familiar with fundamental computing, there's a really good analogy for our words and letters here. If you're typing English in computing in its simplest character-based form, Notepad in Microsoft, there are characters that are available to you, and there is a binary encoding of those characters, right? So you're typing G, but the computer only speaks in ones and zeros, we all know. So it has a strings of ones and zeros that if it sees that, it produces a G on the screen. Or you type a G, it stores it as that binary encoding. That is incredibly close to what life is doing with genes and proteins. The biggest difference is that life has 4 letters instead of 2, binary, and that there are 20 amino acids rather than 26 English letters. And if even that was too much computing, Morse code, same thing. 2 languages, but they're very different. A dot is not a letter. And it kind of matters that they're not, right? And that's why I keep saying, well, DNA, amino acids, different story. Amino acids are a different chemical language. They're the ones that I study, and that's where I can talk about both now and the future with far more authority because it's what my career has studied. I made a very deliberate decision not to study DNA except as a fascinated observer because there was amazing work going on by people far better than me already. And so I chose amino acids because they felt understudied in comparison. So I've told you that in DNA we have hundreds and thousands of alternatives and 2 alphabets. In amino acids, synthetic biology has hundreds, if not thousands, of alternatives and doing much the same classes of phenomenon we talked about for DNA. Same sort of reasons, same sort of motivation. There are no alphabets. And I guess that's where I can finally conclude. What we're doing is working our way towards producing the first alternative amino acid alphabet as a direct sort of comparison, almost an homage to what they've already achieved with genetic material. But for some reason, nobody's gone there. So that's where we're going with collaborators in Japan and Europe, and but we are doing that because nobody else is doing it that we're aware of.
As far as I understand it, the measures for success are to be the best or to be the first.
And let's say that for right, it doesn't matter if you want to be a businessman, but also if you want to be an academic, find an area where nobody's doing it because what you said, right? Be the first, you're the best.
That, or possibly, as in the case for amino acids for life, be the one that survives.
Yeah. I mean, I think that we won't. You want some pure conjecture? I'd say informed conjecture. I think we won't find it hard to make an alternative amino acid alphabet, but I think we have a ways to go studying why those ones are inferior to the one that life has used. Is just where I expect we're going with origin story, which brings us back to economics. All of that is conjectural at the moment. It's informed, but personally, that's what interests me. I would love it if 10 years from now, we might have enough information to conclude whether or not life's amino acid alphabet was a sort of almost inevitable outcome of natural selection. Because again, that says very interesting and important things about our expectations for life elsewhere in the universe.
Amino acids, DNA, RNA, nucleotides, these are all things that, to my A-level understanding of biology, one leads to the other. They are in sequence, they are a family, but it sounds like, to your eyes, they are in competition, almost.
Yes, but I think in a happy competition, mostly. RNA is the sister language to DNA, and the popular paradigm may be changing right now about where life started is it started with RNA, because RNA can do the work of both DNA and proteins. It works like DNA, but it really folds up and catalyzes reactions as if it thought it were a protein. So that's the RNA world hypothesis. Biggest single problem with the RNA world hypothesis is showing how you would make RNA in the absence of life. And this is why I was so strong on that point. You don't find even the building blocks in meteorites. You find the subunits of the building blocks. And the interesting thing here is, but you do find amino acids. No qualifications, no ifs, no buts, no small print. You find amino acids. In fact, you find half of the 20 that are used in you today to make you, and you find a ton of others. But there's half of what makes you you that you don't find, and that's part of an interesting story. And there's a ton of other ones that aren't in you and have never been in life as far as we know it. And that's an interesting part of the story. It is true that if you're a biochemist, one of the decisions traditionally you've had to make is, are you an RNA person or a proteins person? And I think 50 years on, that is part of where they're not talking to each other as much as would be helpful, right? But a lot of the competition is like, I think the way I'm trying to tell it, and it got the excitement out of me, they inspire me. And I wonder if we can't cross-inspire them and actually see it as not competition, but complementarity. Because that's the way life sees it. Proteins are complementary, necessary other half to DNA. And DNA is the necessary complementary other half to proteins. It's the only way we know how to build life.
Well, apart from that, it sounds like you found a third half to put in there.
Yeah, we're just growing both halves. and saying it doesn't have to be this way, and we're increasingly chemically competent to state that as fact. So all those big questions then necessarily tumble to mind. Well, why this one then? Accident? Inevitability? What would be the consequences of doing it otherwise? At least one of the consequences is making a lot of money and owning it. But there are others. Yeah.
Seen Fritz Lang's Metropolis?
No, but I'm so aware of it as the seminal sci-fi work, right?
There is the separation of the intellectual higher echelons, the head, and then the labouring hands, and that the synthoid, the gynoid in the middle, becomes a heart uniting them. And what do you say about growing both halves? It sounds like there is something to be said for the complementarity, but also an elevation, a unification. Lots of words of a very similar
I couldn't
strain.
agree more, and I love the way you put that.
To look at the structure, the classical structure of DNA being a double helix, how much does that topology, that engineering, that shape hold true for synthetic DNA?
I think I would answer in the following order of priority. The number one, this is a field in its infancy, so we don't know. And that's harkens back to this idea of there's so much we don't know in biology. The next thing I tell you is I've really understated a bit because of the DNA side of things, they've gone further. Two fully synthetic alphabets have been produced, which sort of replace DNA altogether, right? Alphabets, yes. Instead of the four that all of life is used for most of its history on Earth, it's a different alphabet. One is known as the Hachimochi alphabet, and that's Steve Benner's group, and you can look it up on Wikipedia. And the other one doesn't really have a catchy name, but I think of it as the Feldman alphabet. Again, you can look it up. I wish it had a name. I think yeah, anyway. That one is actually more interesting to me because if you really want to get into the biochemistry, it's closer to breaking fundamental rules that we've all grown up on. But there are two alphabets at work for DNA. In addition to all of this careful placing of hundreds and thousands of individual modified nucleotides at specific places to do a specific job, right? So that's the state of the field there.
Now, when we say alphabets, is that just a pure like case substitution, a cipher, if you will, for DNA?
Gotcha.
So A is replaced with one or green, like a one in, one out substitution, or like how many rules can you break?
That's the open question. And I mean, does injustice to Benner's group because it was an amazing thing he achieved. But I would say his one is more like the one for one substitution. The Hachimochi is you look at the chemical structures, and if you're trained as a biologist with a bit of biochemistry, at first you think you're looking at life, and then you look, wait, the groups are in the wrong place, right? So it's more like a one-to-one substitution, subgroups within the chemical structure. The Feldman one interests me precisely because it's much nearer to rule breaking. You can totally tune out the next 15 seconds if this doesn't make sense. But for anyone who's listening who does think chemically, the standard DNA and the Hachimochi alphabet use hydrogen bonding to make Watson-Crick base pairs. That's one type of chemical bond. Almost to sort of show off, but in the best possible way, the Feldman one uses something called van der Waals forces, which is what holds graphite sheets together.
What holds geckos on the walls?
Right, right. It's a fundamental rule of biochemistry that has been broken. And I didn't see that coming. So that's why I'm slightly more impressed by that one. No disrespect to Steve Benner, right?
I mean, what you've said there is essentially on a field like it uses magnetism or gravity to hold together. It's that same fundamental universal force.
Yes. And if we dig deeper down, both of them deliberately adhered to the double helix. Partly because they're already going a quantum light year ahead of anything we knew was possible, but partly, I think, because I don't want to mislead you that the current state of play, to the best of my knowledge, meaning there might be things happening in companies I'm not aware of, is that these things are inserted as long stretches within an otherwise normal genome. So you can sort of switch over the genome to the alien language to do your thing. So it's kind of important that they adhere to the basic geometry of DNA. But I think this is where I would see a logical progression, like the ... ellipsis. Benner to Feldman to holy cow, we could make something that was not even a double helix in my lifetime, right?
That's a lot to throw out there in
Okay.
15 seconds.
Oh, you know what? And I need to just say once more, and we aren't talking about amino acids, and there is key differences. So when we get to amino acids, which is what I do and love, but the analogy is so powerful, I'm more than happy to have this conversation with you about DNA. Yep.
Well, to stand with the Feldman alphabet and the Hashimoto alphabet and look back at traditional DNA, your homegrown,
Isn't it fun to use that word with traditional DNA? Right, please take it away.
your organic grass-fed DNA,
Right.
is the state that we find so much of chemistry and life just a matter then of availability, of energy, of economics? If we can establish that these rules can be broken, that the fundamental bonds that hold DNA together don't necessarily have to be hydrogen bonds, then what can we reflect on and say, this must be why life on Earth is the way it is, if there is a
Good.
reason to be found?
I mean, you've nailed it. That's why people like me are interested in all this, even when it's synthetic biology, because we're interested in why is life on Earth? Just full stop. Why is life on Earth? But why is it what it is? Why? And this is a powerful way to get at that, isn't it? Even as a graduate student in the mid 90s, many of my colleagues were respectfully tolerant of the work that my PI was guiding me to do, but felt like these were unaskable questions. There's one example of life, one way of doing it. It seemed a little bit out there to talk about, why and what were the alternatives. And now it is clearly a very good question that most of the early career scientists in one way or another are thinking about. I think your question is the right one. I absolutely believe you've nailed the cause personally, economics. But be aware that that is not yet fully established. I'm just excited by the way that, for me, the clues are pointing at it. In fact, give me one moment of being that philosophical poet. I've come to change the way that I understand what we do in science, and I want to encourage others, if this makes them more interested, to do it. I think I was raised to believe it was about proving things, like bricks on a wall. I think I see it much more now like a form of statistics we call Bayesian statistics, which is more like the odds that you use at a betting course. You use the information you have to guide you to what is most likely to be the current best understanding of the truth. And that's why things change and paradigms shift. But that paradigm has shifted over this issue since I was a grad student. And I would say that right now there is reaching the tipping point of saying life on Earth, DNA, is what it is because economics. Now, a lot of fine detail, which economics, what exactly you're talking about. In fact, I should stop and say that I think that case is much clearer for the amino acids, but then again, that's what I study and can talk about with authority, economics. So we'll get there. Yeah. Okay.
Well, let's go there now, and to say that the case for amino acids, applications, research, and the kind of the synthetic possibilities therein has been a focus of a recent paper, a review that you put out. So if you could talk us through some of the core concept behind that paper, how it relates to your research, the research that your group is doing.
Good. So actually, we've done two reviews, and it's been a January project, 2 years running, except that last year's January project took 12 months to write up. So, yeah. But the first one was exactly what you said. It was a review of synthetic biology in this field for the amino acids, and really making the case that we've made in this conversation that one community really wants to know more about why life is what it is, because that carries very interesting implications for looking for life beyond Earth, as apart from anything else. Another field is excitedly doing this for biomedical and commercial purposes. And that there's relatively low communication between the two groups at the moment. They think differently, they're asking different versions of the question. But it was a paper about trying to sort of cross-educate, if that doesn't sound too condescending, two fields. Oh, by the way, for any young listeners who might go into biology, please, please start to believe how little we understand about each other these days, right? I think this is assumption like I could never do that because they understand everything. Then we fake it, right? And my
I was about
origins
to ask, you
people
mean
don't really.
biologically or just in terms of what the department down the corridor is up to?
I think all academics deep down. I think we have specialised to an extent in an atmosphere where, yeah, we don't often find it easy and vulnerable to admit how much we're struggling to understand a colleague in our own discipline these days. Right? Just a personal opinion, but the only point there was meant to be a positive one to say don't let a lack of understanding stop you from going into this. That's exactly what gets me into it, right? Anyway, sorry, back to the script. Where were we? Commercial people. Sorry, the two reviews. So we wrote the one that was explicitly cross-educating the audience. Last year we wrote one that was much more focused on the origins question, but they do add up to your point. It's saying that we have abundant evidence that life could be other. We have growing pools of data about what would be the consequences of being other. And the time is here to start putting those two together and coming up with answers to questions that seemed unaskable 25 years ago.
That feels, if anything, like not just a light to children, but a simplification to me in the call with you of, like, there's a lot of asterisks and parentheses in that sentence of ways that it could go or not, or different,
Absolutely.
or sideways, or spiral
Yeah.
elsewhere.
Yeah. I mean, it could go anywhere, couldn't it? And I think that part of what we do is believe that the safest place to put as much information as we can is in the public domain. That could be so naive because science can be used for all kinds of purposes. But I know that I trust the public domain more than I trust proprietary companies. I do believe that science, particularly when it's about truth rather than making money, is a uniting force globally. I do regularly converse with people from nations that we aren't terribly friendly with right now. But our conversation never goes there because we're talking about what we're learning about the universe. And that is yet another reason why I'd love to see more people go there. And I do believe, given the amount of money flowing through this for all kinds of purposes, that the companies are doing this. But part of the way the world works is they're not telling us because that's how they're going to make money, right? So, I mean, a lot of what we're doing is trying to put the data and the information out there, and encourage a global community of people interested, hopefully, in truth rather than money per se, to sort of grow this body of knowledge so that we can be informed both scientifically and as as global citizenry about what's going on and what we want to do with it.
Well, in terms of other things that are moving fast and hopefully happening as much for public benefit as possible, synthetic biology, xDNA, all the components that you're working on, you've said already how they lead into mathematics, into chemistry, into computational biology, all of which are undergoing something of a radical change with advents of generative AI, machine learning, database warehouses of DNA. How do you see these two fitting together in terms of stuff that could be happening today, could be happening next, or that anyone listening to this who thinks that maybe they have the same verve for biology as inspired you could lead in their lifetime?
The thing that drives me now for a career, I can say that, in the interface of computers and biology is the untapped potential, not least because computing itself is improving so quickly. But biological knowledge is improving so quickly. I told you earlier in the conversation I'm a bioinformaticist. That's how I'm classified professionally by the university where I work. It's not untrue, but I believe that bioinformatics has come to represent a subset of a bigger, interesting frontier called computing in biology, right? And what I do lies right on the edge of the two, but I want to encourage anyone listening that that is an interface with untapped potential. It is a bit like, the border between the USA and Canada, or the England and Scotland, how you want to pick it. It depends where you're at on the border as to what you're seeing. Could be anything, right? Could be mountains, could be lakes, could be whatever. So it's that frontier, its breadth, its diversity that is so exciting to me. Um, I think that the best sort of grandma advice I could give to anybody considering this for a college and maybe a career is train a little in both fields. Maybe pick one of them to be your major and the other one to be the minor, as the Americans would say, or just train in both. But you don't need to become an expert in both because there's so much untapped potential. The reason to train in both is obvious, but it's what I was just saying. I mean, I say within a discipline, we barely understand each other. Computer scientists don't understand DNA, and biologists don't understand GIGO, right? And there's just so much there, so much to explore and find and discover, and be the first person ever to have discovered it.
From a student retention and recruitment point, we should say biology major with computer
Yeah,
science
we
minor.
should. There we go. There we go. Although I sometimes think that I dream of futures where we stop bothering with that sort of, you've got to be one or the other. Just, yeah, somebody who does both, I'd love. And if we have to teach computer scientists more biology, it's a retooling of our profession, but a very noble one in an age of ChatGPT and that same architecture being applied to no end of sort of biomedical frontiers, right? I'd like more biologically savvy computer scientists. I think that'd be a good thing for the world. Yeah.
Very interesting and important things about the inevitability and possibly predestination of life on Earth.
Absolutely.
For anyone listening to this to find out more, where can they find you on the internet, academically speaking?
We have a humble little lab website, which you're welcome to go to, the Freeland Lab. My university is called UMBC, and it is a great place. So by all means, come and visit UMBC, visit the biology department, and visit our lab website. And you'll certainly have links to our papers there. I did all of, even that was last year, and I'm like, okay, let's do this. And I did all the basic stuff. We're working with a group in Prague who are building our alphabets, and that is the Kluchova lab. I think Europe is ahead of America right now on the sorts of things that I care about. So Kluchova is Hlouchova. That's another great place to visit where they're building stuff in the laboratory to explore the unknown world of proteins, right?
And to round things out, if there's anyone listening to this who is considering a academic switch to bioscience, possibly from computer, maths, geology, or anything else, or they want to get started, is now a good time to get into bioscience?
Yeah, yeah, let's say a market reality is the best time to get into something is when its enrollments are going down because it's supply and demand. They're starting to compete for you. You can get into a better school now for biology because everyone's hurting a bit. So there's a simple reason. Yes, good time. Intellectually, yes, good time. Exciting things are happening. You asked me earlier, what is my dream for 5, 10 years? I do believe that we should find life, independent life, outside of Earth within a generation. Most of my community would say we'd be surprised if we didn't find it within 10 years. The only rider is to say equally excitingly if we don't. That's telling us there's something fundamentally missing from our understanding of the chemistry and history of our planet. So it's an exciting time. Synthetic alphabets, biomedical engineering, genetic engineering taken to the next level on one front, and on the other front, a very real search for signs of life outside of Earth, which the biggest reason when people say, well, why haven't we found it yet? We haven't actually looked. There were 3 experiments in the mid-1970s, the Viking, and one of those came back by its own terms as having demonstrated the existence of life on Mars. But it was explained away as we hadn't understood soil chemistry at that point. So, point is, we haven't looked. So I think 2 different reasons why this is intellectually a great time, and the market says, yeah, get into biology right now because they'll fight to get you. Right? One of the ways to start sewing them together is by saying we reach a point of common interest when we say it could be other. Those people are inspiring me more than the people who study detailed genetics these days. I mean, I struggle to keep up. But I know a lot about the synthetic DNA because it inspires me. And so thank you for that. And, maybe we inspire a generation here, or at least a few individuals, to be the next steps in sewing those 2 back together again.
That'd be nice.
That would be
to
nice.
imagine. I don't know if anyone is listening to this. We're recording it in the early days of 2024, but, should we all still be here in 2030, then drop us a line and let us know. For now, I'll say, Professor Freeland, thank you so much for your time.
Thank you.